it's important to regulate the osmolarity of your body. So essentially, how much water do you have relative to the solutes, like sodium and proteins and calcium and other things? And regulating that is called osmoregulation. All organisms have to do this, but the challenges are different for different kinds of organisms depending on where they live. If you live in fresh water, then you have more solutes in your cells than there are in the environment. And water is constantly going to be diffusing via osmosis into your cells, and you have to pump out fresh water. If you live in the desert, you're going to be losing fluid frequently. Or if you are a marine animal living in a very salty area, it can desiccate your body quickly, deplete you of water, and so you have to conserve water or get rid of solutes. And excretion is getting rid of nitrogenous metabolites and other waste products, and this often goes together with your regulation of osmolarity, because you're essentially regulating what's in the fluid in your body and what's not. So osmoregulation is regulating the osmolarity of the fluid inside your body. But what is osmolarity? Well, it's the solute concentration of a solution, and that determines the movement of water. If you have more solutes on one side of a membrane than the other, then that is hypertonic, right? And water is going to move in that direction. If you have the same amount of solutes on both sides, you are isoosmotic or isotonic, has the same tonicity, and water is not going to move in either direction in net. So in this case, if this membrane is not permeable to these solutes, but is permeable to the water, this side is hyperosmotic or hypertonic to this side, and water is going to move in this direction until you even out the concentration. Hopefully you guys remember this fairly well from Bio 131. There are some organisms who do not have to regulate their osmolarity. We call them osmoconformers. These are mostly marine animals that live in very, very stable osmotic environments in terms of places in the ocean that have a very specific osmolarity, and so they don't have to regulate it, right? Because it's not going to be changed by their environment. But most organisms are osmoregulators, meaning we expend energy to control our water uptake and water loss. Those osmoconformers tend to be marine invertebrates. Vertebrates in the ocean, and some invertebrates, and everything not in the ocean tends to be an osmoregulator. Marine bony fishes tend to be hypoosmotic to seawater, which means they are going to lose water to the sea, and they're going to gain salt through osmosis and from food. And so they're going to have to balance that by drinking seawater and excreting salts, using energy to do those two things. So bringing in seawater and then pumping out the salts to maintain their consistent osmolarity. Freshwater animals are in a hypoosmotic or hypotonic environment, meaning they are hyperosmotic or hypertonic to their environment, which means water is going to come in. And so they are going to lose salts by diffusion, gain water by diffusion. And so they're going to have to replace those salts with food and uptake across the gills. So marine water fish and freshwater fish have exactly opposite osmoregulation challenges. And this is why most ocean fish can't live in fresh water and most freshwater fish can't live in the ocean. Some invertebrates live in temporary ponds. And so when those ponds go away, those ephemeral ponds evaporate, they're able to go into a stage called anhydrobiosis, where they essentially live in a dormant state that needs almost zero water. They're incredibly water efficient because they don't do anything until water comes again, and then they come out of this anhydrobiotic state and become normal again. One excellent example of an organism that does this is the tardigrade or water bear. This is how they look when they're hydrated. This is their anhydrobiotic state when they don't have water around. They're very, very, very cute when they are hydrated. For land animals, the key challenge is avoiding water loss because you're not sitting in a body of water, especially if you live in a dry area like a desert. And there are lots of different ways, both anatomical and behavioral, that organisms that live on the land will avoid water loss. A lot more animals, for instance, in the desert are active at night when it's not as hot and you're not going to lose as much water through evaporation than during the afternoon. If you go out in the desert on a summer afternoon, nothing is going on. It's just empty and quite unpleasant. And so land animals have to maintain water balance not only by losing as little water as possible, but by consuming as much water as possible, by eating moist foods, drinking water from ponds, and producing water metabolically through cellular respiration. All of this, of course, does not come free. If you are an osmoregulator, you have to expend energy to move salts or to move waters one way or the other across a membrane, right? You have pumps that require ATP to function because you are moving some substance up its concentration gradient, right? And that always requires ATP. And because it's expensive, you don't want to just conserve water for the sake of conserving water. Right? You want to conserve only as much water as you need to conserve. One example of this is your nitrogenous wastes. So all animals have nitrogenous wastes from the breakdown of proteins and nucleic acids. And those things tend to be converted to ammonia, NH3. And ammonia is toxic. And so you don't want it just sitting around in your body. You need to excrete it. And if you are a fish, that's fine. You can just secrete it as much as you want with water. So essentially, fish use water to dilute ammonia, and they can use plenty of water because they're living in it, right? And so this is why you have to change the water frequently or have a good filter on a fish tank if you have fish in your home. Otherwise, it'll fill up with ammonia and it will kill the fish. That'll happen in a fish tank, but not in the ocean or a large lake or pond. So that's why they're able to just leave the ammonia as it is. Mammals and most amphibians and even some fish change ammonia to urea. Urea is less toxic than ammonia. It's not totally non-toxic, but it's less toxic than ammonia. And so it requires less dilution, less water to dilute. So this saves us water. We couldn't, get rid of as much water as a fish does per our body size to dilute ammonia and still be able to maintain our hydration in a terrestrial environment. We would dehydrate very quickly. So urea, converting ammonia to urea is expensive, but it allows us to conserve more water. If you live in a drier area, and this includes a lot of insects and reptiles, and that includes birds, because birds evolved from reptiles. If you either live in or evolved from an organism that lives in a dry environment, you may spend the extra ATP to convert urea to uric acid, which is even less toxic. And so that way, you hardly have to dilute it at all with water. And so while it requires a lot of ATP, it does not require a lot of water to get rid of it, essentially. And so reptiles, birds, a lot of insects produce uric acid, urine, essentially, that doesn't come with a lot of water, so they're able to conserve water. The organs or body structures that animals have evolved to regulate our osmolarity and to get rid of nitrogenous wastes are fairly different between different animal groups, but they usually involve some complex of tubules. One of the oldest and simplest is called a protonephridium, and nephro usually refers to kidneys, so this means like before the kidneys. It's a network of tubules that dead end that are connected to some external openings, and they have this cellular unit called a flame bulb, and that allows for some exchange. Here you can see an example of a protonephridium in a platyhelminth, so a flatworm, and this is all your tissue fluid, and it's drawn into these tubules because there are cilia in this flame bulb here that draw fluid in. You have some filtration here, and then this tubule, right, sends the fluid, this is kind of like their urine, out the opening body wall and into the environment. Earthworms have something a little bit different and slightly more complex called a metanephridia, which is a series of tubules that collect coelomic fluid. This operates in a fairly similar way to your protonephridia, except in addition to the flow of fluid and that's sent out the external opening, along the way, in addition to sort of this flame bulb, you have lots of exposure to capillaries so that you can have exchange and you can reabsorb some of those solutes that you don't want to get rid of. So you can hold on to that stuff. But you can leave the stuff you do want to get rid of in the filtrate and you can send it out. So they leave in, for instance, urea. And it's fairly dilute what they're getting rid of because they have to keep getting rid of water because they tend to live in moist freshwater kind of habitats. This looks a lot more like what we'll see in a nephron, which is the part of the kidney that mammals use to excrete. Most terrestrial arthropods, like insects, do it a very different way. They have something called a Malpighian tubule. And this does not involve that filtration step where you're sort of pushing water through a filter. Instead, you essentially pump solutes across into the place that will be kind of like the bladder where you're going to get rid of the urine, and water follows, but then you reabsorb that water back into your body, and really all you have is some dry waste matter that's primarily uric acid, because a lot of these insects evolved in dry areas. So here you can see these tubules where you are actively pumping salt, nitrogenous wastes into here that will come into essentially the rectum. And then water follows through osmosis, but we reabsorb that water from the rectum until you get this kind of solid waste that's mostly, again, nitric acid. Now this is a relatively energetically expensive process, but it's really, really good at conserving water. In mammals, our organs of both excretion and osmoregulation are our kidneys, and they're found, kind of in the upper end of your abdominal cavity, but in the back here, and they have a cortex on the outside and a medulla on the inside. And if you take part of this medulla, which is called a renal pyramid, and the cortex that's outside of it, this is one lobe, one renal lobe. Renal refers to kidney. And if you zoom in, each of these has lots and lots of nephrons. And nephrons are the functional unit of the kidney. It's what actually does the excretion and osmoregulatory function. Some of these nephrons are primarily in the cortex. They just dip into the medulla a little bit. We call those cortical nephrons. That's most of them. Some nephrons are a little bit in the cortex, but most of the tubule is down in the medulla. We call these juxtamedullary, or next to the medulla, nephrons. And in one particular nephron, you can see that blood comes in to this place here through an afferent arteriole. Afferent means ingoing, efferent means outgoing. So the afferent arteriole brings blood in to this ball of capillaries called a glomerulus. That's a fun word to say, glomerulus. And this ball of capillaries is made of capillaries that have little slits in them, little holes. And so you have a bunch of blood pressure, and then a bunch of stuff squirts through these holes. It's like if you put a balloon, like a water balloon, on the end of a hose and turn the hose on to fill it up, but you put a bunch of holes in the water balloon, right? Water is going to squirt out everywhere from those holes. That's what's happening here. That's filtration, like what we saw with the protonephridia and metanephridia. And what goes through is not just water, but anything small enough to fit through the holes. That means not cells, those are too big, not proteins, those are too big, but small things like urea, for instance. We want to get rid of that. But also things like sodium and calcium and glucose, which seems important, but we get rid of it through this filtration process. Anything that's too small. And size isn't always the best way to decide what's important to your body. So after that, we have a series of tubules. A proximal convoluted tubule that's closest to this, glomerulus. A nephron loop, and then a distal convoluted tubule that's farthest away from the glomerulus in terms of flow of filtrate. And then these are all surrounded by blood capillaries, and that allows for some exchange to occur. It means that small things that we want to keep in our blood, we get to pump back. All that glucose we pump back because that's fuel. We don't want to waste it. A lot of the sodium, depending on how much sodium we have in our blood, or calcium, we may want to pump a lot of that back. We may not. It depends. And a lot of that water we want to pump back because we want to stay hydrated. And it also allows us to pump the large stuff that we want to get rid of out. Sometimes we have toxins, for instance, that are too big to leave through filtration, but we want to pee out anyway. Things like, the Tylenol that you just had, last night for your headache. And that gets pushed out, pumped into the filtrate from your blood because it was too big to go out during filtration, but it ends up in your urine anyway, and it ends up from the distal convoluted tubule into this collecting duct, and that collecting duct will collect all this urine from different nephrons and put it into the main part of your kidney where you can urinate it out.